got you on this page, I even pointed at the compare region viewer, but we're still not going to look at that yet. The first thing we're going to do is create some data groups that we can compare in the viewer when we get there. So one of the things that I would like us to do is to create some feature groups, which are proteins or genes, and then some genome groups. So you notice that here under functional properties, we've got the BV-BRC local families or global families. And also we have identical proteins and identical genes. You may be wondering, what are these local and global families? Well, those are all based on this publication, which are the PADI FAMs. And these are the reasons that we think it's important for you to annotate your genomes in the BV-BRC system, because every bacterial genome that comes through gets an assignment of a PL FAM and a PG FAM. So PL FAM are the local families, meaning they are within a genus, and they're based on Kamer assignments. And the global families are cross-genome families. So if you wanted to compare something within a specific genus or species, you'd probably want to look at the local families, the PL FAMs. And if you wanted to go broader and do cross comparisons across different genera, you'd want to do the PG FAMs or the cross-genus families. So going back here, you see that we have the local families and the global families. So I'm going to copy this, and I'm going to show you kind of a quick way. If I were to just click on this, and you'd see it be a really huge number of genomes. Yeah, 201, which is a lot for the viewer. So let's try to make it just so it creates a pretty picture of these protein families that are class A beta lactamase genes. They're important in the resistance to ampicillin. I'm going to show you a different way to search for things. Certainly you could do it this way and create a feature group of this. And in fact, we can do it there. You notice if I click here, it selects everything, and I can go here to the group, and it says it's a feature group, and it says, is this a new or existing group? Well, this is a new group. So I click the down arrow, click new group, and I'd say, I'd put the PL family ID, all proteins. So there, that's pretty simple. And that gives me this message telling me that it's created this group. But I want to show you a different functionality that we have in BV-BRC. And you can go up here into searches and click on that, and let's click on proteins. And so I'm going to do the taxon name, Haemophilus. And so I have to click on that. And then down here, these are just ways you can scope your search to be really specific. Here under genome length, I want the complete genomes. I don't really want the whole genome sequence. I want the genomes where the number of contigs match the number of chromosomes, and you've got everything there. And down here under additional protein criteria, we click on this down arrow, and then I would click on the PL family ID and enter that in, and then do the search. We'll just wait for it to load, and let's see how many of these it gets. And you can see it gets 28 from a bunch of different Haemophilus genomes. So we would click on this, click on the group in the green action bar. Remember, we want it to be a new group, and do this and say complete. Now, just for some variation, because often things within the same genus will have constrained or similar gene neighborhoods, I may want to look a little bit broader to try to see where in the evolutionary time frame that things start falling apart and we see this gene in different contexts. So I've got the PG fam. Remember that PG fam is the cross-genus family. So I'm going to look a little bit broader. So I'm going to go into proteins here, And this time I'm going to do the past EU. I want the family, Pasteurella this family of bacteria to which Haemophilus belongs. I want those to be complete too. And this time, clicking on the down arrow, I want it to be the global family. So I'm going to click on that, put that in, and click search. And you can see it returned 30 results. There's a lot of Haemophilus here, but as you can see here, we have a couple of Pasteurella. So it'll be interesting to see how those guys look compared to the Haemophilus. I think it'll be interesting. Who knows, it might be boring, but we'll find out together. So let's click on this checkbox and it selects all 30 of them all together. And then remember, in the green action bar, I go into group and I want to create a new group. And it's this guy for the global family, and I'm putting complete there. I create a lot of groups. It's important to me to give them names so I can remember what they are when I see them. Okay. Want to do something a little bit different now. I want to create genome groups, and we don't have to be at the same place we were before with that particular gene, although that is going to be where we're looking at the compare region view. So let's click on this one row, one particular gene, and I don't know anything about this gene. I don't care that much about it, but it's going to provide me with a gateway to get to the genome group I want. So here, I'm going to click on the G icon for the genome icon. So I click on that, and it opens up not the feature landing page, but the genome landing page. And so this is all the information about this particular gene. But I want to show you something really cool that we have in BV-BRC. So up here across the top, this is a breadcrumb, and each of these are a hyperlink to a landing page for each of these entities. So let's click on Haemophilus influenzae. Here, this is the information about this specific species. And you can see there are this many genomes, and we have this information on the antibiotics that they are susceptible, which are the green color, or resistant to, which is the red color, or intermediate. Now, the beta-lactamase gene is important in ampicillin resistance. So we want to create two groups, one that's susceptible to ampicillin and one that's resistant to ampicillin. Now, I could get there by clicking on either of these. And in fact, let's just do that right now. So I click on this, and this is showing me that there are 72 genomes in the resource that the isolates were found to be susceptible to ampicillin. So I'm going to click on this to select them all. Also, let me point out that you can see that it was by broth dilution, a laboratory method. And actually, let me click this off. If I click on an individual one, you can see that it's got a PubMed ID. And you can even see the paper of that if you click here beyond the green bar. When you click on a single genome, it gives you additional information about that genome beyond the green bar. So if I click on this, it should provide me with a hyperlink to PubMed. And that paper from 2019 by Pinto et al. Looked at Haemophilus influenzae, and they recorded the antimicrobial resistance. And now I have to, of course, do this. So I can pop back here. Then I'll do this again because the picture's prettier when you see it all. And I'm all about providing you guys with the prettiest pictures. So I'm going to click on this, select all of them. And this time in the group icon, I'm going to create a genome group. So let's click on that. And I want it to be a new group. And I'm going to call it AMP Haemophilus. All right. So we've got this group. We click it, and then we look down over here, and it says it's created it. Now, we were initially here on the overview. So I could click on the resistance here, but I want to show you something we can do here, which is the AMR phenotypes. So let's click on that. And that shows us all the data we have for all of these genomes. Look at the PubMed ID here. I'm going to click on this again and double click. I could see that there were two different IDs in here. So there are a couple of papers that are describing ampicillin resistance and susceptibility within Haemophilus influenzae. But let's click on the filters here. And this time I want to choose the antibiotic, which is ampicillin. And this time I want the resistant. Now there are only 16 genomes here, but that's okay. Let's click on this and let's create a group. And then it's a new group. And I'm going to call it AMP resistant Haemophilus. Okay. And there it says we've popped it up. And if you've been working on these things, or let's say you created groups a while ago, or you just created a group and you can't remember how many genomes were in that, you can go up into the workspaces, and then I can click on genome groups. And so that's going to show me all the groups I've created with the two I created most recently, which is just today. And here they are, and this one has 72, and this one has 16. And if I wanted to do feature groups, I could click here. And so it's showing me the PG FAMs and the PL FAMs. Oh, and look at this. I've got all of these together. So there are three here. I don't often show this, but this is the Venn diagram function. And it's going to show you how many genomes are in each of those. So let's click on the Venn diagram. And that shows me for this particular group, it has two unique ones. And this, remember those Pastorella? Those are those guys. And here are the PL FAMs, all protein, the PL FAMs, the complete, there were 28 that they share and 73 that are different. And if like you wanted to see that particular group, you could even create a group of that by clicking on it. How cool is that? We don't often show the Venn diagram function, but I love it. It's one of my favorites. Okay, we've seen all of this. We've created our groups. Now we're getting ready to actually see the viewer. I know you can hardly wait. And in the next video, the secrets will be revealed. I'll see you there. Bye.